Hello everyone. Today we are going to speak about our next lecture, that is genetics part 9. And today we will directly start with what we had stopped in the previous lecture. So in the previous lecture we had stopped at dihybrid laws. So now I will continue with the laws which we had to discuss. Okay? So today the law which we will discuss is law of segregation. Law of segregation. Law of segregation is also known as law of purity of gametes. Purity of gametes. Okay, now what is this law? Okay, let us speak about this. Now consider if there is a heterozygous organism and I am crossing two heterozygous organism. Okay, then we know that if this is capital T dominant allele and this is a recessive allele, if these two allele they separate means to segregate. Okay, segregation is also known as separation. Separation is also, segregation and separation are the same words. So capital T and small t, these two are segregating, means they are separating from each other. If they are separating from each other, then this is what each is known as gametes. This is gametes formation. When there is a pair of factor, like here the pair of factor you can see, this is the pair of factor. What is this? Pair of factors. Okay, if there is pair of factors, these this factors, what they will do, they will separate from each other and they will enter into the gamete formation. Means what, they will form different gamete. It is not like that capital T small t this is one gamete. No, they are forming separate gametes. Okay, like one is capital T which will give you a tallness effect. This gamete small t will give you the effect of dwarfness, right? Each gamete will give its own effect. Hence we say that gametes are pure. Because now why they are pure? Now why I will say that pure? Because it's showing only one thing at a time. If it is a capital T allele, it's carrying only one allele, that is known as capital T. Okay. If it might have carried capital T small t, then this is not pure and this will not occur also. Okay. So it is carrying only one allele. How many allele it is carrying? Only one allele, and this one allele will give one effect. So we say that gametes are pure. Okay, what we say that gametes are pure as they will carry only one allele. Either it will carry dominant allele or dwarf allele. Dominant or recessive. Your dominant is tall, okay, and dwarf is recessive, right? So it will either carry dominant or recessive allele. So it will carry only just one allele out of those two alleles. Earlier there was two alleles, so it will carry only one allele. This two alleles will segregate. It will separate to form different gametes. So we say that gametes are pure, and hence this law is known as law of segregation, or we say law of purity of gametes, and this law is a universal law. Okay, what type of law it is? Universal law. Yes, so this is about the second law of genetics, known as law of segregation. Okay, now if we move ahead and we speak about another law, third law of genetics, which speak about the dihybrid cross. Okay, we remember that the dihybrid cross was there, and dihybrid cross was giving us the ratio, a phenotypic ratio as 9 is to 3 is to 3 is to 1. So a dihybrid cross, it was giving you a phenotypic ratio. Which, what was the phenotypic ratio? Phenotypic ratio was 9 is to 3 is to 3 is to 1. This was the ratio which we had got. Okay, so we can go like at that cross actually. We had taken something known as like this. You know, if you remember, we had taken, uh, let us say about the F1. F1 was something like this. Yes, there were two different characters considered. There was a character for seed shape, there was a character for seed color, right? So if we represent it on the chromosome, so it will look like this. We took capital R, capital Y, r, small y. So if this was the scenario, okay, then the chromosomes actually, you know what happened in this case, this there was a separation of chromosomes, and chromosomes separate independently. How? See, there was one possibility of we getting this as one gamete. So the chromosomes are separating in this way, you are getting this as one gamete, then we are also getting this as one gamete. We are not saying that capital R, capital Y, small y, this is as one gamete. No, this is not a gamete. Gamete formation occurred this way. This is one gamete, that is capital R, capital Y. This is one gamete, that is small r, small y. We got another gametes also. By crossing over, we got capital R, small y, and we also got small r, capital Y as the gamete. So we got capital R, small y, capital Y, small r. We got four gametes. So these gametes of two characters, how many characters are there? There are two characters. Now, one character is for seed shape and another character is for seed color. Okay, one character is for seed shape and another character is for seed color, right? And these two characters, they independently separated. Okay, they separated independently. So there was separation. Independently they separated. Independently they separated. Okay, independently they separated. So after the separation, like, okay, they separated, fine, they separated, but after separation they assort independently. Now what is meant by assort? Assort means to distribute among themselves. Okay, to distribute among. Okay, after, we can say after separation there was assortment. There was assortment. Assortment means distribution among. Okay, means what actually happened? There were two pairs of characters, right? two pairs of characters, and each pair of character had contrasting factors, contrasting allele. If there was round round seed shape, there was wrinkled seed shape, these were two contrasting characters, they like contrasting traits, so they separated, right? And even if there is capital Y, small y, this also independently separated. There was no dependence among anybody in this case. So each pair separated independently. Okay? And hence we got the ratio as 9 is to 3 is to 3 is to 1. So this ratio came because there was independent separation first thing, and they distributed, means among themselves they distributed independently, so there was independent assortment. Okay, so this is known as a law, which is called as a law of segregation. Okay, law of independent assortment. So segregation was here, this is independent assortment, law of independent assortment. Okay, it's independent assortment. Okay, so this is about the third law of genetics. Yes, so we have completed law of dominance, law of segregation, and law of independent assortment. These three things were done already. And before that, we have also spoken about monohybrid and dihybrid cross, back cross, and test cross. So we are done with almost, you can say, 30 to 40% of the chapter here. Let's go forward and we will understand that there is another topic known as deviations from Mendel's findings. This is also known as Neo Mendelism. Neo Mendelism. Same topic, neo. Neo means what? What research, like what experiments were done by the Mendel on the green, like all the pea plant. Those experiments, those findings, when somebody else had done the same experiment, so they got something different, they got some other results, so there was slight change, or you can say deviation. Deviation means what? When we speak something from the exact thing, like suppose Mendel law is a standard thing, so we got something, some other result from the standard. Okay, let us say that if it was 1 is to 2 is to 1 ratio of phenotypic cross, okay, so we got some other ratio, maybe 1 is to 2, something else, some other ratio. So that is known as deviation from Mendel's findings. Whatever Mendel had searched, there was something different which people got, and hence this is known as Neo Mendelism. And this was like this all occurred in the era when Mendel was not there. So it was post Mendelian era. We say it as post Mendelian era. So let us begin with one by one. So what Mendel told, that if there is one single trait, okay, if there is single trait, then there will be single gene. Okay, if there is single trait, then there will be single gene, and each gene has two alleles. Okay, if there is single trait, there will be single gene. Single trait will have a single gene, and that will have two alleles. We can also say that one gene is having two alleles, right? Me now during that time when like he also told that gene he that time the word genes was not there, so he told it as a factor. Okay, so even Mendel thought that if there are two alleles, so these two allele will, out of these two alleles, like if we consider that there is a gene for height, okay, and there are two genes for it, one is capital T and small t. So out of, Mendel told that out of these two alleles, only one will be dominant, only one will be dominant. This is what Mendel told. Okay, and even he told that there will be independent assortment. Okay, they will like independently assort. These were the points which Mendel told, which we had already studied. But what happened, there are some, some deviations. Okay, so there are exceptions to the first law of genetics. There are some exceptions. Now what are these exceptions we will study in this part. So there are total, we will go with the four exceptions of this Mendel laws, first law of Mendel, that is law of dominance. So we will first understand that what is intragenic and intergenic interaction. This is very important. Now you will understand that why this is important, because of the interaction between genes. Okay, now this is intragenic interaction and there is intergenic interaction. So what will happen, because of the interactions between the genes, you will get some different effects. Say intra means what? Intra means if suppose there is capital T, small t, so there will be interaction between alleles of same gene. If there is, this is one gene, right? One gene for height of a plant. So there will be interaction among these two alleles. So interaction is there between the alleles of the same gene. That is known as intragenic interaction. And due to this interaction, there was a deviation which occurred in the first law of genetics. And a deviation, there are two examples of this deviation. One is incomplete dominance, and the second one is known as codominance. Second one is known as codominance. Now we will study this all one by one. Okay, so because of these two alleles of the same gene, they were interacting among themselves. Okay, so there was interaction between these two alleles of the same gene. Because of that, there was a cause known as intragenic interaction, and there were two deviation. One is incomplete dominance and the second one is codominance. Okay, speaking about intergenic. Intergenic means non-allelic. Okay, now intergenic means there are two different genes. Okay, let us say there is gene known as capital R small r and capital Y small y. This is a different gene and this is a different gene. So whenever there is interaction between the two different genes, okay, if I consider one chromosome, and if I say that this is a gene, okay, I will color it. This is one gene of orange color, and there is another gene of light pink color. So if these two genes are interacting among themselves, then we say it as non-allelic genes, because this is another like these two are same alleles of one gene, and these two are same alleles of one gene, but between these two there is no allelic, like we can't say that this is allele of the same gene. No, because the genes are different, so we say non-allelic. Okay, So here, if there will be interaction between two different genes, so we say it as intergenic. But suppose if I take red color, and if I say, suppose if there is this, and here is one allele, like, okay, this is the same gene. Let us say this is the same gene. It has two alleles. And if the interaction is only in this one gene between the two alleles, then it is known as intragenic interaction. So the example for intergenic interaction, there are two examples. One is known as pleiotropy. Okay, and we are going to study about pleiotropy. And then you have polygenes. Okay, we have polygenes, but it is not there in your syllabus. And there is epistasis. Again, there is no topic given in your textbook about epistasis. Or again, there you have supplementary gene, and you have complementary genes also, like complementary genes interaction. So these all topics, out of these all topics, your textbook mentions about pleiotropy only. Only pleiotropy they are mentioning. Rest all topics they are not mentioning, so we are not going to speak about those. Okay. So this was all about the two laws of genetics which we studied. Law of segregation. Then we studied about dihabit, like the basics of dihabit, which we had already done. Then this is second law. Okay, means third law of genetics. In this video it was the second law. And then I spoke about these interactions. Okay, if the interaction is between only one gene, if I consider a very huge chromosome, if interaction occurs only in one gene, then this interaction will be intragenic. Okay, between these you have two alleles. Okay, one gene has two alleles. And if there is interaction between two different alleles on same chromosome or on different chromosome, then this interaction is known as non-allelic interaction or intergenic interaction. So we will come towards the next part, that is incomplete dominance. Incomplete dominance. So in incomplete dominance, what happens, that we have let us directly come to with the examples. So here one plant, uh, the flowers were taken of mirabilis jalapa, mirabilis jalapa. Okay, this is a scientific name. And this flower was taken, this is a flower, right? This flower was taken, it had two colored flowers. One was red colored and one was white colored. So there is red flowers, the phenotype, and here there is white flowers as the phenotype. Okay, so what will happen, you will get gametes. So since these two are similar, I will take only capital R. These two are similar, I will take only smaller. So there will be fusion of gametes, you will get capital R small r as F1. Now, since we know that law of dominance spoke that only dominant allele will express in F1. So according to that law, F1 must be all red. Okay, what should be the F1 must be all red. But instead of showing red color, it didn't show red color. This actually showed a pink color. Okay, so these were red flowers, and these red flowers are dominant. Okay, these are dominant. And these white flowers, okay, the white flowers, which is, this is a white flowers I'm highlighting with gray. These white flowers are actually recessive. Okay, what are they? They are recessive. But in F1, the dominant red capital R didn't appear. There was an appearance of pink color. Pink color was appeared. And if you go with the F1, selfing of F1, you know what is selfing? Self pollination of F1. Then we will get capital R small r crossed with capital R small r. And if you solve this by your monohybrid, like the way we solve monohybrid cross, we get here two gametes of each. Okay, if we do this all, we have already done how to remove these things. So I'm directly writing the answers first. So if you see this, so here they came to know that capital R is red. Okay. But capital R small r is pink colors. And this small r, it is white color. So the phenotypic ratio in the findings, the phenotypic ratio, it came as 1 red, okay, 2 pink, okay, and 1 white. This was the phenotypic ratio, that is 1 is to 2 is to 1. And speaking about the genotypic ratio, the genotypic ratio was, if they got genotypic ratio as 1 capital R and 2 capital R small r and 1 small r. So here the case was such that the genotypic and the phenotypic ratio was, like both ratios were coming as 1 is to 2 is to 1. So in this case 3 is to 1 was not the case for phenotypic ratio. But when we studied that in monohybrid cross, we got that the phenotypic ratio should come 3 is to 1. But here we are not getting as 3 is to 1. So this is a deviation from Mendel's experiment. So this is exception of monohybrid cross, or we can say exception to law of dominance, because law of dominance told that in F1 generation only the dominant will express, but here neither the dominant is expressing nor the recessive is expressing. There is something which is a middle character, that is pink color is coming as a phenotype. So we say it as incomplete dominance. What we say? Incomplete dominance. Neither the dominant expressed nor the recessive. Nobody expressed here. Neither dominant nor recessive. Nobody expressed completely. Nobody expressed. Neither R told that yes I will express red. No, this also didn't happen. Neither small R told that I will express white. No, nothing of this sort happened in this case. Nothing happened. Nobody expressed. So we say it as incomplete dominance. Neither dominant expressed nor recessive. Okay, so in F1 all are pink. Okay, this is important. In F1 all are pink, and hence we got the ratio as 1 is to 2 is to 1. So this is about incomplete dominance. So this is entirely about incomplete dominance which we studied, and it will be a question asked in any entrance exam, that what is the, which, like we can say which, okay, which which cross has both phenotypic and genotypic ratio as same? Which cross has same phenotypic and genotypic ratio? So which is this cross, if this is the question? So the answer is, there is a cross known as independent. Oh, I wrote something different. It's incomplete dominance. And this is giving the number as 1 is to 2 is to 1 for both phenotypic ratio. PR means phenotypic ratio is also same, and GR means genotypic ratio is also same. For both the ratio there is 1 is to 2 is to 1, and this is important for sure. MCQ question in any entrance exam it can come. Okay, it can come for sure. Which plant was taken in the experiment? It can also be a MCQ question here. I am highlighting it. It's Mirabilis jalapa. This can also be an important question. It can also ask you that in F1 which color, which phenotype was asked. This can also be an important question, that pink color was observed, and this can also be MCQ. They can ask that whether which is Mendel's Neo Mendelism topic. Okay, or they can ask what is deviation from Mendel's law. So this is a deviation known as incomplete dominance. This is also deviation, but deviation from which law? Law of dominance. Okay, this is incomplete dominance is a deviation of law of dominance. So this can be also a question. Okay, and you should remember the this phenotypic ratio, they can give you this in the match the following, and they will ask you that what is the phenotypic and genotypic ratio. So this is also an MCQ. So I hope there are, you can see that so many MCQ are there in this topic. If you want to know about MCQ, see in law of segregation they can ask which law is, they can ask you that which law is a law which is universal law. So this MCQ can be asked because first law of dominance is not universal. Okay, if it might, if it would have been universal, then we won't would have got this incomplete dominance. Okay, every time it should have been law like dominant. Every time F1 should be dominant, but this we didn't got in this. So law of dominance have exceptions. What it has? It has exceptions. And the very first exceptions means it is what? It is not universal. Not universal law. This can also be a MCQ question. Okay. So we had taken this all, right? You should know the examples of which is intragenic inherit this interaction. Okay, you should know it is intergenic interaction. You should know this all. Like here the examples, you should know that incomplete dominance is intragenic interaction. Codominance is also intragenic interaction. You should know that polygenic, polygenes, epistasis, supplementary, complementary, and this pleiotropy. These all are intergenic interactions. Again, this can be asked as MCQ here. Okay, right? This can be asked as MCQ, which I am telling that what is non-allelic. They can ask whether epistasis is allelic interaction or non-allelic interaction, then you should know that Epistasis, as I have written here, that epistasis is non-allelic interaction. What is epistasis? Non-allelic interaction. What is supplementary? Non-allelic interaction. What is complementary? Non-allelic interaction. You should must be able to say this. And inter intragenic is allelic interaction. Okay, it is allelic because it is in between the same gene. Okay, they can ask the definitions of this. So these are the expected questions which I am telling you. Okay, it is expected question from you. So I hope till here we are understanding the concepts. Next we have to study about the codominance, and then we have more topics. So codominance, multiple alleles, pleiotropy we will take in the next lecture. With this we will come to the another topic known as the chromosomes. We will start because chromosomal theory of inheritance we had already taken in the beginning of the classes. So after this we will do three topics from the deviation. Then I will start with chromosomes in the next lecture. Okay, and if there is chromosomes, after chromosomes there is linkage. Whole part of linkage is done. Crossing over is also taught. Directly after these three topics and chromosomes I will come to Morgan's experiment. Okay, so this is the three topics, and then we have autosomal and sex chromosome inheritance. Okay, sex chromosome interactions will be there. Okay, so there will be, I will just write inheritance here. Okay, let me mention as a whole. Chromosome inheritance. So you will have this, and lastly we will have sex determination and genetic disorders. And then you have genetic disorders. So you have how many topics total? Total you will have topic 1, 2, 3, 4, 5, 6. So here what are the three topics we will study? Codominance. Then we are going to study about multiple alleles. After multiple alleles, we are going to study about pleiotropy. Okay. And after pleiotropy, we are going to study about this. So what I will do, how many lectures it will take more, I will tell it now first. See, so for these three topics and chromosome, we will take one class till here. So there will be one class for this for sure. So there will be lecture number 10 in which I will take these four topics. There will be lecture number 10, then I will take 11 where you will study these two topics. Okay, so there will be a second class, and lecture number third you will get this. So there will be more three lectures about the theory, and I will take fourth and fifth lectures as MCQ part. So till here I will stop the class for now. We have done these all concepts to understand. Okay, and do like, do subscribe, do comment, and keep studying. Thank you.